Hello and welcome back to Zoology 141. Today's lecture is going to be on nervous tissue. Okay, today's lecture on nervous tissue will actually be the first of probably 2 or 3 different lectures on the nervous system. Today we're going to look at the basic processes and the anatomy of nervous tissue, whereas in future lectures we'll look at the central nervous system, peripheral nervous system, and finally move on to talk about the specialized sensory receptors we have, the eyes, ears, nose, etc. And so the functions of the nervous system can be divided into one of 3 very broad categories. First of all, there's a sensory function. The nervous system receives input from sensory receptors, such as our eyes, our ears, and even the temperature receptors in our skin. And so sensory input is one of the 3 main functions. The central nervous system, that is the brain and spinal cord, does something called integration. It takes this sensory information that has come into the CNS and it analyzes it and interprets it. And finally, the last function of the nervous system is to generate a motor output. And a motor output is just nerve impulses traveling away from the brain or spinal cord going to some sort of effector organ. And an example of an effector organ would be skeletal muscle. Remember, our brain is what tells our skeletal muscle to contract. But other examples of effectors could be things like endocrine glands and exocrine glands. So this shows a very simple mock-up of how the nervous system works. Basically, sensory information comes in from sensory receptors, for example the eyes. It travels to the brain or spinal cord where integration takes place, that is analysis. And finally, if necessary, the brain or spinal cord will generate an appropriate motor output for that particular input. In this case, that motor output is going to be affecting skeletal muscle. So the nervous system is actually divided into 2 main categories. The central nervous system, which consists of the brain and the spinal cord, and everything else. Everything else here is called the peripheral nervous system, and it includes the peripheral nerves and nerve receptors. And so the peripheral nervous system can be divided into a sensory side and a motor side. The sensory side is where our sensory receptors are located. Again, eyes, ears, nose, and even the sensory receptors in our skin. So nerve impulses traveling from the sensory area travel towards the CNS, and remember the CNS is where that sensory data is going to be analyzed and interpreted. On the other hand, the motor division of the peripheral nervous system is where the output comes from the central nervous system. That is, if we need to contract a skeletal muscle or have a gland secrete something, that will be the function of the motor division of the peripheral nervous system. And so the motor division here is actually subdivided into 2 different categories, the somatic nervous system and the autonomic nervous system. The somatic division mainly controls skeletal muscles, so this would be the division that controls voluntary contraction of your biceps muscle, quadriceps muscle, and so on. On the other hand, the autonomic division controls involuntary body functions. That is, things like heart rate, blood pressure, things that your brain and spinal cord are controlling via motor impulses, but which you don't have conscious control over. And finally, the autonomic nervous system itself is subdivided into parasympathetic and sympathetic divisions. And these are 2 very antagonistic divisions, meaning they have opposite effects on, let's say, heart rate and blood pressure. One will speed up heart rate, for example, the sympathetic nervous system, and one will slow it down, the parasympathetic nervous system. And so we'll have a separate lecture on the autonomic nervous system in just a couple more weeks, but you should know that the motor division is divided into the somatic division, which again controls voluntary muscle movement, and the autonomic division, which controls involuntary body functions. So nervous tissue is pretty cellular. That is, it consists mainly of cells and not a whole lot of extracellular tissue. And the cells here include neurons and neuroglial cells. Now, neurons are cells that we've talked about before, and they're specialized for communication. They generate special action potentials, which are essentially impulses that are electrical that travel long distances throughout the body. On the other hand, neuroglial cells are sort of the accessory cells. They don't generate electrical potentials or charges, but they do provide support and protection to neurons throughout the body. And examples of neuroglial cells include things like Schwann cells. Now your book goes into great detail about the anatomy of a neuron. Remember, the neuron's function was to generate electrical charges that can travel long distances throughout the body in order to communicate. And so the main parts of the neuron include the cell body, the dendrite, the axon, and then also the myelin sheath. So the cell body is the area where the nucleus and the majority of organelles are located. Attached to the cell body are structures called dendrites. Now, dendrites are special receptors that receive nerve input from surrounding neurons. Most neurons are talking to several other neurons, and they do this through axodendritic connections. Neurons also have an axon. An axon is a long conducting portion of the neuron which conducts the nerve impulses away from the neuron cell body and towards the synapse. It's important to realize that the action potential or nerve impulse is actually generated at an area called the axon hillock, which is just between the cell body and the axon proper. And finally, the axons of many neurons are covered with something called the myelin sheath. The myelin sheath is just an insulating layer which is composed of neuroglial cells. And we'll talk about the function of this insulating layer in just a few slides. So we can classify neurons in a few different ways. First of all, we can classify them based on their function. And based on their function, there's three different types of neurons: sensory neurons, interneurons, and motor neurons. So sensory neurons are neurons that carry information from the external environment, that is from the skin, from the nose, from the eyes, to the central nervous system, the brain and spinal cord. And so the sensory neuron here is shown in blue. It's bringing afferent information that is going towards the central nervous system. On the other hand, interneurons are special neurons that are found only within the central nervous system, and their job here is to integrate and process information that's been provided by the sensory division. And finally, the motor neurons are neurons that take information away from the central nervous system and bring that information towards effectors, for example, skeletal muscle or glands. And so the purpose of the motor neurons is to generate an appropriate motor output based on the integration that's going on inside the central nervous system. And so motor neurons are often said to be efferent because the action potentials are traveling away from the central nervous system and to other structures in the body. Another way we classify neurons is based on their shape or their anatomy. Now, the most common neurons are something that are called multipolar neurons. These neurons have three or more processes. That is, they have one axon and at least two dendrites. Usually they're going to have lots and lots of dendrites. And so the multipolar neurons are very common, comprising over 99% of the neurons found in the nervous system. The second type of neurons are called bipolar neurons. These neurons have only two processes, one axon and one dendrite, and these processes extend away from the cell in opposite directions. Now, these neurons are quite rare and are found mainly in the special sense organs, for example in the retina of the eye and also the olfactory epithelium of the nose. The last neuron type is something called a unipolar neuron. These neurons have a single short process that emerges from the cell body, and then it divides into a T-junction, and that T-junction splits off into a distal peripheral process, and then it also has a more central process that enters the central nervous system. Unipolar neurons are found mainly in the peripheral nervous system, and they're very common in the dorsal root ganglia, which is where the sensory information comes into the spinal cord. So a lot of sensory information will travel into the central nervous system via these unipolar neurons. Now that we've talked about neurons, we need to go on and talk about the neuroglial cells. Remember, neuroglial cells were accessory cells that are part of nervous tissue, but the big difference between them and neurons is they do not actually generate any type of action potentials or electrical charges. And neuroglial cells include things like astrocytes, microglia, ependymal cells, and oligodendrocytes. These are the neuroglial cells found within the CNS, or central nervous system. Remember, that means brain and spinal cord. So within the central nervous system, we have things such as astrocytes. Now, astrocytes' jobs are to anchor neurons to the capillaries and also guide connections between neurons. On the other hand, microglial cells are there to scavenge up any type of dead or dying cells and also to mop up pathogens. Essentially, when a pathogen is detected, these microglia can transform into something that's sort of like a macrophage and scavenge up the pathogens as well as dead cells and debris. Another type of neuroglial cell found within the central nervous system is something called ependymal cells. The ependymal cells line cerebrospinal fluid-filled cavities within both the brain and the spinal cord and have a role in generating this special fluid. And finally, the last type of neuroglial cell found within the central nervous system are the oligodendrocytes. The oligodendrocytes are the cells that are responsible for insulating the axons of CNS neurons. So they wrap around and form something called the myelin sheath, which is very important for speeding up nerve impulses and also protecting the axons. Now, within the peripheral nervous system, we find fewer types of neuroglial cells, but they are still very important. And these include things like the satellite cells. Now, satellite cells surround the neuron cell bodies that are located in the peripheral nervous system, whereas Schwann cells are the cells responsible for forming the myelin sheath within PNS neurons. Now, the myelin sheath is very important both in the central nervous system and also in the peripheral nervous system because it helps to speed up the velocity of nerve impulses. Another thing that it does within the peripheral nervous system only is that these neuroglial cells can help to repair and regenerate damaged axons. For example, if you happen to sever a peripheral nerve, it is possible that that nerve can regrow those connections because of the special guidance provided by the Schwann cells. But this cannot happen within the central nervous system. If you happen to sever a tract or cell axon within the central nervous system, that axon is going to stay severed and probably that neuron is going to degenerate and die. So within the CNS and PNS, we can have both myelinated and unmyelinated fibers. Now the myelinated fibers obviously are either oligodendrocytes in the CNS or Schwann cells in the PNS that are wrapping around those axons. And again, they're there to help speed up the velocity of the nerve impulses. And we'll talk about how that happens in just a few slides. We do have some unmyelinated axons, however, and they tend to be slow and have very small diameter axons or fibers, and they're only surrounded by something called a neurolemma, but they don't have a myelin sheath. And because they don't have a myelin sheath, their nerve impulses tend to be fairly slow. So just remember, we find myelin both within the central nervous system and also within the peripheral nervous system. In the peripheral nervous system, the myelin is generated by our Schwann cells, whereas in the central nervous system, the myelin is generated by our oligodendrocytes. Now the myelin is a fatty type of material, and so if you look at it macroscopically, it tends to look a lot whiter than the surrounding tissue. And that's why in the brain and the spinal cord, we can differentiate white matter from gray matter. The white matter contains primarily myelinated axons, so it's just a bunch of cables that have this special myelin covering. On the other hand, gray matter is an area where we have the cell bodies, and we also have, let's say, synapses between adjacent cells, but very little to no myelination, and that's why it tends to be a lot darker than the surrounding white matter. So just remember, in the brain and spinal cord, when you see gray matter, that's areas where the cell bodies are located, where synapses are occurring, and everything else, the white matter there, is basically just electrical cables or axons traveling from one region to another. So now that we've learned a little bit about the anatomy of both the neurons and the neuroglial cells, we're going to go on to talk about how information is transmitted along neurons. So information within a neuron is transmitted by an electrical charge, which is called either an action potential or a nerve impulse. And this action potential is actually a temporary reversal in charge from negative to positive. Normally, the inside of an axon is a little bit negative, and during an action potential, it briefly turns positive. In the next few slides, we'll talk about how this process occurs. Now, before we go on to talk about nerve impulses, which are action potentials, I should point out that, in fact, there are two different types of charges that can occur within a neuron. The first one of these, which we've kind of already talked about, is an action potential. And this is a temporary reversal in charge from negative to positive that travels all the way down the axon of a neuron. And this is the way that neurons use to communicate with one another and also to communicate with and control effectors, for example, muscles or glands. On the other hand, graded potentials are electrical charges that are isolated just to the cell body and the dendrites, and they don't travel down that axon. Basically, if you think about action potentials and graded potentials in terms of the human sexual response, an action potential would be an orgasm, whereas a graded potential would be a stimulus that is just caused by foreplay or something like that. It doesn't necessarily lead to orgasm, but with enough stimulation, enough graded potentials, that eventually can summate and lead to an action potential. So I do apologize for using what some people might consider a crude analogy, but this is one that I found that's worked pretty well in communicating to students the differences between graded potentials and action potentials. So action potentials potentially have 3 to 4 different phases. These include a resting phase, depolarization, repolarization, and hyperpolarization. Now during the resting phase, a neuron is negative on the inside, and we'll talk about why that is in a few slides. If it's stimulated enough, we go through something called depolarization, and during depolarization, the charge inside the axon will switch from negative to positive. During repolarization, those charges flip-flop back again. The inside of the axon becomes more negative. And finally, we sometimes go through something called hyperpolarization, in which the neuron becomes super negative, and because it's super negative, it tends to make it less likely that it's going to generate another action potential very quickly. Now this slide just shows a graphical presentation on what's going on inside of an axon both before and after an action potential or nerve impulse. So first of all, take a look at the left-hand side of the graph. You'll notice that the baseline of an axon, that is the normal resting voltage, is around negative 70 millivolts. Remember, a millivolt is 1/1000th of a volt, and so it's a very, very tiny electrical charge, but one that's very important. And we'll talk about why it's a negative charge in just a few slides. Suffice it to say that if we stimulate that neuron, either mechanically or sometimes chemically, we can get the resting voltage to increase, that is, become a little bit more positive. It goes from negative 70, let's say, to negative 65. And if we stimulate it a little bit more, it can go from negative 65, let's say, to negative 55. And negative 55 is a very important voltage because this is the threshold. This is the point at which the graded potentials will summate and become an action potential. Again, if we're thinking about the sexual response analogy, this would be the point of climax or orgasm. And so once we reach threshold, we are going to have an action potential. And the first part of that action potential is depolarization. The charge inside the axon rises rapidly, surpassing zero, and it becomes positive 40 millivolts. So that happens because of influx of sodium ions, which we'll show in just a few slides. On the other hand, repolarization is the opposite process. Here, the charge goes from positive 40 back down to a negative charge, and this is a result of potassium ions leaking into the cell. And finally, after repolarization, we're going to have something called hyperpolarization. Hyperpolarization is just having a charge that's more negative than normal. Normally, we have a resting charge of negative 70 millivolts, but sometimes after an action potential, it drops down, let's say, to negative 90 millivolts. And that puts us into something called the refractory period. Now, the refractory period is the period in which a neuron is not likely to form a second action potential because it's a lot further to get up to threshold. So in case you're wondering, I do consider it very important that you be able to draw and reproduce this graph on an exam because it will show that you understand the 3 or 4 processes that are going on during an action potential. So in the last slide, we looked at a graphical view at what's going on with a charge inside an axon during and after an action potential. Now we're going to go through those 3 or 4 steps more with a pictorial view. So here we can see the resting potential of a neuron. And remember that resting potential is negative, specifically negative 70 millivolts. If that neuron is stimulated adequately, we will then have the action potential occur. And the first stage of the action potential is something called depolarization. Now, during depolarization, the inside of the axon becomes positive because we have sodium ions that are coming into the axon. And relative to the inside of the outside of the axon will be negative during depolarization. Now, during repolarization, we restore the initial resting potential. That is, the axon goes from being positive back to its original negative charge. Now we're going to look at the 3 or 4 stages of an action potential or nerve impulse in greater detail. Here we're concentrating on what's happening in the axon and outside the axon. That is, what's making it negative and what's eventually going to make it positive. So first of all, let's take a look at the resting potential. Now the resting potential, as we said earlier, is going to be negative. That is, the inside of the axon is more negative than the outside of the axon. But let's talk about why it's more negative. Well, first of all, inside the axon we do have proteins, and these proteins do have a partial negative charge, which already gives the axon a little bit of negativity. The other thing we have is something called the sodium-potassium pump. The sodium-potassium pump is a membrane protein that uses ATP or energy to force ions against their concentration gradient. And specifically what it does is it pushes sodium to the outside of the membrane, that is in the extracellular space, and potassium inside the membrane, the intracellular space. And the way that it works is essentially 3 sodium are pumped out for every 2 potassium that are pumped in. Now you might say that both of these ions are positively charged, and so how does that result in a charge difference across the membrane? But think about this. If I'm pushing 3 positives outside for every 2 positives that I'm putting inside, the inside will become relatively more negative than the outside, which is relatively more positive. And so this resting potential is maintained by the continuous pumping of the sodium-potassium pump. Now before we go on to the next slide, I want you to think about where these ions would like to go based on diffusion. Sodium is initially a lot more concentrated outside the axon, so it wants to come inside. And potassium is more concentrated inside the axon, so it would like to go outside. Just keep that in mind as we move on to the next slide. Now the second part of a nerve impulse is depolarization. Now remember that depolarization is a rapid reversal in the charge inside the axon. It was negative and all of a sudden it becomes positive. So why did this happen? Well, take a look at what happens. During depolarization, we have special membrane proteins that are specific to sodium which open up. And if we have enough of these open up, this allows a lot of sodium into the axon. And the sodium moves into the axon because of diffusion. It was a lot more abundant outside the axon and virtually non-existent inside the axon. So it very quickly moves through the membrane proteins and down its concentration gradient. And this temporarily changes the charge inside the axon from negative to positive. Now during repolarization, the sodium gates will close and the potassium gates will open. Remember, potassium was initially more concentrated inside the axon, and so it tends to rush out very, very quickly. Now as potassium rushes out, it brings the charge inside the axon back into the negative range. And so this is called repolarization because it brings us back down to the resting membrane potential. And finally, once the depolarization and repolarization have occurred, the resting membrane potential will be restored by our sodium-potassium pump. That is, it'll take all the sodium that was inside the axon and very quickly pump it to outside of the axon. And it will take the potassium that was outside and pump it to the inside. And so these membrane proteins are always expending energy to keep these ion gradients the way they are. Sodium more concentrated outside the cell and potassium more concentrated inside the cell. Now, as I said at the beginning of lecture, I think it's very important that you understand how the process of nerve impulse transmission works. That is, you understand depolarization, repolarization, and the resting membrane potential. So make sure you go back and study those last 3 or 4 slides so you know why the axon becomes negative and positive when it does. There's also a link here to a very good and simple animation that shows the process of an action potential. So be sure to visit this animation before you go on to the next slide. So now that we know how an action potential occurs, we're going to talk about the manner in which it travels across an axon. One very common misconception that students have is that the action potential or depolarization happens simultaneously across the axon, and that's not true. In truth, it actually travels like a burning fuse. That is, it starts at one point and moves sequentially to the end point of the axon, called the axon terminus. So let's take a look at the transmission of a nerve impulse in closer detail. So here we have a portion of an axon that is undergoing an action potential. You can see that depolarization is happening on the left part of the axon where sodium is rushing in. But take a look at the right part of the axon. The inside is still negative, indicating that everything else downstream is still in the resting membrane potential. That is, it's negative inside the axon. But that depolarization will spread in a wave-like fashion from the axon hillock down to the axon terminus. So now we can see what's happened. The action potential has spread or moved down the axon to where the middle part of the axon is depolarizing, whereas the left part of the axon is now repolarizing. Potassium is rushing out in order to restore the negative charge inside the axon. But you can also see that the right part of the axon is still in its negative resting phase. and finally that depolarization will reach the end of the axon where sodium will rush in, causing depolarization there. Meanwhile, more proximally, we'll see that repolarization is happening in the center of the axon, and finally the proximal part of the axon that was attached to the cell body is already back at its resting membrane potential. So bottom line here is that the action potential or nerve impulse travels sequentially down an axon, much like a wave at a football game. So now we're going to talk about the way in which the nerve impulse travels down the axon. And this will vary depending on whether or not that axon is myelinated. Non-myelinated axons use a type of conduction called continuous conduction. And during continuous conduction, the depolarization travels down the axon sequentially, like we saw just in the last slide. On the other hand, saltatory conduction is a type of conduction that occurs in myelinated axons. Here it's important to realize that the depolarization only happens in the spaces between the adjacent neuroglial cells. And so we'll see a better representation of this in the next slide. So this slide just shows a comparison of continuous conduction on your left and saltatory conduction on your right. Remember, continuous conduction is a type of conduction that happens in non-myelinated axons. Basically, depolarization starts at one end of the axon and travels sequentially down the axon until it reaches the axon terminal. And this takes quite a bit of time. On the other hand, take a look at the type of conduction that happens in myelinated axons. Now, myelinated axons are insulated by either oligodendrocytes in the CNS or Schwann cells in the PNS. And these cells are discontinuous, meaning there's a gap in between the adjacent neuroglial cells. And these gaps here are called a really funny name, and that name is the nodes of Ranvier. Ranvier, I guess, was a physiologist, and that's back when you used to name things after yourself. And so he described these gaps in between adjacent neuroglial cells, and that's why we know them as nodes of Ranvier. So it's important to realize that the depolarization in myelinated axons only happens in the nodes. It doesn't happen in the insulated portions. And so it starts at one end, jumps to the next node, and then the next node. And as a result, saltatory conduction tends to be a lot faster than continuous conduction. So just a summary about what we learned in the previous slide. We have two different types of conduction: continuous conduction that occurs in unmyelinated axons, and saltatory conduction which occurs in myelinated axons. In general, saltatory conduction tends to be a lot faster because we're only having a certain number of depolarizations across that axon. So myelinated axons tend to be faster at transmitting impulses than non-myelinated axons do. Another factor that affects the speed or velocity of nerve impulses across an axon is the axon diameter. In general, larger axons tend to be much quicker at transmitting impulses than smaller diameter axons. And so we divide our nerve fibers or axons into three different categories: Group A fibers, which have large diameter myelinated axons; Group B fibers, which are intermediate in diameter and only lightly myelinated; and finally Group C fibers, which are small unmyelinated fibers. Now the Group A fibers, because they're large and myelinated, transmit nerve impulses very quickly, around 150 meters per second. Whereas the Group C fibers that are unmyelinated and very small in diameter transmit nerve impulses at a paltry 1 meter a second. That's very, very slow. And so remember, more myelination speeds up the nerve impulse, as does axon diameter. So now that we know that neurons communicate via action potentials, we need to talk about how they differentiate between different types of stimulus intensity. Now I would like you to think about two different types of tactile stimuli, that is, stimuli we can feel with our hands. So first of all, on the left-hand side of the screen, you can see that we have somebody touching a feather. So close your eyes, think about what a feather would feel like. Now the fact that you can feel the feather and are aware of it means that there's enough stimulus there to initiate nerve impulses or action potentials that are traveling towards your brain. And your brain is where the interpretation of that stimulus takes place. And that interpretation says, oh yes, that is a feather. On the other hand, let's take a look at the stimulus on the right-hand side of the screen. Let's imagine that you're out nailing a board or something like that, and you accidentally slam your finger with a hammer. Now, because you feel that pain or are aware of it, obviously that means we have action potentials or nerve impulses that are traveling to the brain and spinal cord. Of course, you probably realize that these nerve impulses are not pleasurable. They're actually very painful. But how does your nervous system differentiate between these two different types of stimuli? Now, one common misconception that students often have is that, if we have a very heavy or painful stimulus, the action potential must be much greater in magnitude. But that's not actually the case. Whether we are touching a feather or whether we have slammed our finger with a hammer, the actual magnitude of the action potentials tends to be the same. So what's the difference? In most cases, our nervous system communicates the differences in stimulus intensity based on the frequency of action potentials. That is, both touching a feather and being slammed by a hammer generate nerve impulses. The difference here is that the more intense stimulus, slamming your finger with a hammer, generates much more frequent nerve impulses. So we have a lot more of them, and they tend to be more closely spaced. So this is one way that your sensory receptors can tell your central nervous system about the difference in magnitude of the stimulus just by adjusting the frequency of the nerve impulses. Another factor that we can use to differentiate between a light stimulus and a heavy or hard stimulus would be the number of sensory receptors that are stimulated. If you're just touching a feather, you're probably only stimulating the very superficial sensory receptors that are in your epidermis and maybe the upper dermis. On the other hand, when you slam your hand with a hammer, you're generating a lot more force, and that's activating deeper and deeper sensory receptors. And so it tends to activate many more sensory neurons than would a light stimulus. And so this is another way that your nervous system can use to differentiate a light stimulus from a heavy stimulus. Okay, so now that we understand that the way in which neurons transmit information across an axon is through an action potential, we're going to go on to talk about how one neuron can talk to another neuron. And in most cases, one neuron will talk to another neuron using something called a neurotransmitter. A neurotransmitter is just a special type of chemical produced by neuron A that has an effect on neuron B. And we already talked about the neurotransmitter acetylcholine in the previous chapter. That was the neurotransmitter that motor neurons use to communicate with muscle cells. Now, before we go on to talk about neurotransmitters and so forth, we need to go over some important terminology. And these are two terms, a presynaptic neuron and a postsynaptic neuron. So first of all, let's talk about what a synapse is. Remember, a synapse is where a neuron and a muscle cell meet, or in this case, a neuron and another neuron meet. Most neurons in the body are connected to several other different neurons. And so we'll have an upstream neuron called the presynaptic neuron, which is talking to a downstream neuron, which we call the postsynaptic neuron. The presynaptic neuron is the cell that initially generates the action potential and communicates that information to the postsynaptic neuron via neurotransmitters. Now, based on the neurotransmitter coming across, that postsynaptic neuron is going to have to decide what to do. Does it fire off its own action potential, or does it not fire off an action potential? In order to understand what will happen, we need to know something about the frequency of stimulation and also the type of neurotransmitter that's crossing the cell. So this picture just shows a mock-up of the different types of connections that nerve cells can have with one another. So here we see a neuron cell body. We'll call this the postsynaptic neuron that's receiving information from three different presynaptic neurons. The one up top has an axon that is branching off and synapsing with the cell body of our postsynaptic neuron. And because the axons here are synapsing with the cell body, we call that the axosomatic synapse. Remember that axo means axon, and somatic here means body. So this is a synapse where an axon meets with a cell body. On the other hand, we can also have an axodendritic synapse. This is where the axons of one presynaptic neuron synapse with the dendrites of the postsynaptic neuron. And finally, we can have something called an axo-axonal. That is where the axons of the presynaptic neuron synapse with the axon of the postsynaptic neuron. This slide just shows a summary of the events that occurred in a chemical synapse. Again, we can see the presynaptic neuron on the left side of the screen and the postsynaptic neuron on the right side of the screen. Initially, we have an action potential traveling down the axon of that presynaptic neuron. Now, once it reaches the end bulb, it will cause an influx of calcium into the axon bulb. This influx of calcium will then cause the release of vesicles of neurotransmitter. This neurotransmitter will diffuse across the very narrow gap between the two cells, which is called the synaptic cleft. Once it crosses the synaptic cleft, it will bond to special receptors in the postsynaptic cell. Now, depending on the type of neurotransmitter, this can cause the postsynaptic cell to depolarize or form its own action potential, or it can cause it to hyperpolarize or prevent further action potentials. And so we're going to talk about this in greater detail in just a few slides. So we said a couple slides back that a neurotransmitter is a chemical produced by neuron A that has an effect on neuron B. Now, there are lots of different types of neurotransmitters. To date, we've described more than 50 different kinds of neurotransmitters. In truth, most neurons make more than one neurotransmitter. It can make 2 or sometimes 3 different types of neurotransmitters. And the effect of these neurotransmitters will differ depending on the location of the neuron and also the frequency of neurotransmitter release. We tend to classify neurotransmitters based on their structure, that is what kind of chemical they are, and also their function, that is, are they excitatory or are they inhibitory. Probably the best known neurotransmitter is acetylcholine, abbreviated ACH. Acetylcholine was the first neurotransmitter to be identified, and thus it's the best understood. It is released at the neuromuscular junctions, which we talked about previously, and also within some neurons of the autonomic nervous system. Now, acetylcholine is synthesized from acetic acid and also choline by an enzyme called choline acetyltransferase. This isn't really important, but what is important is that it's degraded by an enzyme called acetylcholinesterase. Acetylcholinesterase is a very important enzyme because it breaks down the residual acetylcholine remaining in the synaptic cleft. And this is very important because when the first neuron sends information to the second neuron, usually we want that message to be very short-lived, because things might change. We might want to turn off that message. And if that acetylcholine keeps bouncing back and forth between those two cells, that message is going to keep echoing and echoing and echoing. And so for this neurotransmitter, we have an enzyme that very quickly breaks it down so that it's not repeatedly stimulating the postsynaptic cell. In addition to acetylcholine, we have another class of neurotransmitters called the biogenic amines. And these are divided into 2 classes, the catecholamines and also the indoleamines. The catecholamines are based on the amino acid tyrosine, and they include things like dopamine, epinephrine, and norepinephrine. Now, epinephrine and norepinephrine you've probably heard of, because not only are they neurotransmitters, but they're also hormones. These are the hormones that we inject into somebody that might be undergoing cardiac arrest or, let's say, having an allergic reaction. They tend to amp up the effects of the sympathetic nervous system. Dopamine, on the other hand, is one of those feel-good neurotransmitters. When you're doing something that's pleasurable, let's say eating a bar of chocolate, we have release of dopamine and similar neurotransmitters. Indoleamines, on the other hand, are neurotransmitters that are based on the amino acid histidine or tryptophan. And the 2 examples here include serotonin and histamine. Now, serotonin is probably a neurotransmitter that you've heard about before because we have certain drugs called SSRIs, or selective serotonin reuptake inhibitors, which basically increase the amount of serotonin residing between the 2 cells, that is within the synaptic cleft. Because we understand now that serotonin has a role in depression and other types of mental illnesses. So we have people that are chronically depressed and aren't responding to other types of therapy. We can give them an SSRI, which will basically prolong the residence time of serotonin within the synaptic cleft between 2 nerve cells. And because it resides in that cleft longer, it stimulates the postsynaptic cell more frequently and tends to reverse some of the or lessen some of the different symptoms we have associated with depression. So the neurotransmitters that we looked at in the previous slides could be both inhibitory or excitatory. It basically depends on where they are secreted within the nerve system. On the other hand, the neurotransmitters that we're looking at now are either going to be one or the other. They can't be both. And a good example is GABA. GABA is an amino acid neurotransmitter, and it's principally an inhibitory neurotransmitter. It's found within the central nervous system. GABA is a neurotransmitter that tends to make us more drowsy, less alert, and we can simulate the effects of GABA by taking, let's say, alcohol or benzodiazepine drugs, for example, Valium. So anything that binds to GABA receptors is going to make a person less aware, less conscious, more drowsy. Now, another type of amino acid neurotransmitter is glutamate. Glutamate is an excitatory neurotransmitter that is found both within the brain and spinal cord. It's important in forming memories, and it's also important in learning. Another type of inhibitory neurotransmitter are something called endorphins. Endorphins are special types of neuropeptides, so they're based on amino acids. And you probably already are aware that endorphins are our natural painkillers. When secreted, they tend to reduce our perception of pain. And so endorphins, for example, are released when you're exercising a lot. You start to feel a lot better at some point during a long run because you have this massive surge of endorphins. Now, the effects of endorphins can be mimicked, of course, by using drugs like codeine or hydrocodone, Vicodin. All of these are opiate drugs that also reduce pain. Our next class of neurotransmitters are the purines, and purines are neurotransmitters that are based on the breakdown products of nucleic acids and nucleotides. For example, ATP, adenosine triphosphate, can actually act as a neurotransmitter. Finally, it's important to realize that even some things like gases can serve as neurotransmitters. One very common example is nitric oxide. Nitric oxide is a gas that is secreted by some neurons, and it can be either excitatory or inhibitory. For example, nitric oxide is secreted by some neurons, causes dilation of smooth muscle cells within arteries and veins. And this in part allows for the filling of erectile tissue during the male sexual response. And finally, the last group of neurotransmitters are the endocannabinoids. Now, you probably heard of these before because it sounds a lot like cannabis. Remember, cannabis is marijuana, and the principal ingredient in marijuana is THC. And so THC mimics some of our endocannabinoids. And endocannabinoids have various roles in the nervous system. They're involved in memory, appetite control, and as well as nausea and vomiting. And that's why in some areas THC or marijuana are actually prescribed for people that have problems with nausea and things like that. On the other hand, endocannabinoids, including THC, can have a deleterious effect on memory. People that tend to smoke a lot of marijuana have some short-term and later long-term memory loss. So as you can see from the previous slides, there are lots of different types of neurotransmitters in the body. The big question is what will that neurotransmitter do to the postsynaptic cell, that is the cell that is receiving the message? Well, that depends on whether the neurotransmitter is excitatory or whether it's inhibitory. Now, excitatory neurotransmitters are neurotransmitters which will cause that postsynaptic cell to partially depolarize. That is, it will go from a negative resting voltage of around -70 to something that's higher than that, let's say -50. So it gets it closer to the threshold, closer to the point at which it might fire off its own action potential. On the other hand, an inhibitory neurotransmitter is one that will cause hyperpolarization within that postsynaptic cell. It was at -70, but once that inhibitory neurotransmitter travels across, it now hyperpolarizes and becomes, let's say, -90. Now, if you permit me to use the sexual response analogy here once again, I can give you a different explanation of excitatory and inhibitory neurotransmitters. So think about things that put you in the mood, whatever that is. You know, for some people it might be a bottle of wine, some soft music, anything that gets you closer to the point towards climax. So that would be analogous to an excitatory neurotransmitter. On the other hand, think about things that get you out of the mood. These would be things that make you think, oh, no way, I'm not going to do that. And these would be analogous to inhibitory neurotransmitters. So, you know, if your boyfriend comes home, brings you flowers and wine, that would be an example of an excitatory neurotransmitter. Whereas if all of a sudden he gets in the bed and belches or farts or something like that, that's going to be a turnoff, sort of like an inhibitory neurotransmitter. Now this graph just shows the electrical potential within the postsynaptic cell once it's been exposed to an excitatory neurotransmitter. Remember, an excitatory neurotransmitter is one which is going to cause the postsynaptic cell to become more positive. And that's what we see here. Its resting voltage is -70, and all of a sudden, bloop, it bumps up to nearly, let's say, -57 or -56, right near threshold. And we call this temporary partial depolarization of the cell an EPSP. EPSP stands for excitatory postsynaptic potential. And it's important to realize that this is a localized phenomenon. It's a graded potential that only occurs in the dendrites or parts of the cell body. It's also fairly short-lived, but it tends to be longer-lived than, say, an action potential would be. Without further stimulus, this EPSP is going to go back down again to baseline unless we have more stimulation. On the other hand, an IPSP is an inhibitory postsynaptic potential. During an IPSP, the postsynaptic cell becomes more negative because it has received an inhibitory neurotransmitter. And this inhibitory neurotransmitter causes the postsynaptic cell to hyperpolarize or become more negative. Here you can see on the graph we're at -70. Here our stimulus arrives, and then we hyperpolarize to, let's say, -80 or -90. And because we're hyperpolarized, it's going to be much less likely that stimulation from an excitatory cell is going to get us up to the threshold, which is still -55. Now, it's important to realize that both EPSPs and IPSPs are graded potentials that occur within the postsynaptic cell. They don't have any effect on action potentials unless they summate on one another. If we have several postsynaptic potentials that occur within very close proximity of one another, these temporary charges can build on one another to eventually get us up to threshold and cause an action potential. So summation here can be classified as spatial summation or temporal summation. Now, spatial summation results when a single postsynaptic neuron is stimulated by multiple presynaptic neurons giving the same message. For example, here you can see is our postsynaptic neuron labeled C, and it's receiving excitatory neurotransmitters from both neuron A and neuron B. Now, here you can see the effects of these neurotransmitters on the resting membrane potential. A little bit comes from A, we become a little bit depolarized, then a little bit more comes from B, a little bit depolarized, and then A and B together, all of a sudden those two waves summate on each other or build for us to reach that threshold of -55 millivolts. And in that case, our postsynaptic neuron is going to go yippee and have its own action potential. A second type of summation is called temporal summation. Temporal here means time, and temporal summation happens because a presynaptic neuron will stimulate a postsynaptic neuron multiple times. For example, take a look at the graph below. You can see that the presynaptic neuron stimulates the postsynaptic neuron once, twice, and then several times in quick succession. If it stimulates it several times in quick succession, these EPSPs, or excitatory postsynaptic potentials, can build up in the postsynaptic cell until we reach threshold and then have an action potential. So in summary, there's two different ways that summation can occur. We can have temporal summation, where a single presynaptic neuron stimulates the postsynaptic neuron several times in quick succession, and we can also have spatial summation, receives excitatory neurotransmitter from multiple presynaptic neurons. Either way, this allows us to get increased numbers of EPSPs and build us up to that threshold potential where the postsynaptic cell can form its own action potential. So this concludes the lecture on nervous tissue. Now, we didn't cover everything in the chapter in this lecture, so be sure you do read your textbook. In particular, look at the different types of neural circuits and neural processing. Also, I would like you to be familiar with the disorders that affect nervous tissue, for example, multiple sclerosis and cerebral palsy. You should also read the Closer Look section located on page 418 of your textbook entitled Pleasure Me, Pleasure Me. This describes the effects of various types of neurotransmitters and neurotransmitter mimicking drugs on the cells of the central nervous system.